Hey, boys and girls. It is a snowy day at Murray, and I was thinking about you all and how you might be a little anxious for tomorrow's test and for the midterm exam that's coming up. So I thought on this snowy day, I would actually send you the lecture via video. So the first thing you need to do is get warm. As you can see, I am. Then get yourself some hot chocolate. Make sure you have good hot cocoa. Oh my God, that is good. And once you have that, you should get ready. The first thing we're going to do is question of the day. Okay, here is the question of the day, number 44. What are the three components of a nucleotide? Okay? Why don't you go ahead and pause this while you write down the three components, and while you're at it, why don't you try to string them together, the three components, into a nucleotide? Okay, so pause it, go ahead, do it right now. Okay, so if you got the question of the day correct, you would have said there were three components: a phosphate group, a pentose sugar, and a nitrogenous base. Now, the importance of nucleotides is that they are the building blocks of nucleic acids. And you will remember there are two types of nucleic acids. Can you remember what those nucleic acids are? Go ahead, take a guess. Say it out loud in your head right now. You're right. They are RNA and DNA, which only differ by the sugar. In RNA, it's ribose, and in DNA, it's deoxyribose. Now, nucleic acids are polymers of nucleotides. And the nucleotides are strung together like beads on a necklace by dehydration synthesis. We've seen that reaction several times. And you can see how that looks in the next slide here. You can see that we have a phosphate, a sugar, a base, attached to a phosphate, a sugar, a base, attached to a phosphate, a sugar, a base, a phosphate, a sugar, a base. So this whole structure here is a nucleic acid. One thing to notice about this nucleic acid is the fact that it is phosphate to sugar to phosphate to sugar to phosphate to sugar to phosphate to sugar, with these nitrogenous bases flapping out to the side. Okay? There are two families of nitrogenous bases. There are some that have a single ring, and there are others that have a double ring. The ones that have the single ring are called pyrimidines. And the two pyrimidines that we find in DNA are called cytosine and thymine. You're going to need to know those names. The two purines in DNA are called adenine and guanine. So they're abbreviated by their initials A, G, C, and T. We'll get to uracil in a little bit, but as you can see here, it is only found in RNA, and it takes the place of thymine in RNA. Once again, I told you earlier that the two sugars are different. In DNA, there's deoxyribose as the sugar. In RNA, it is ribose. You can look very carefully, and you will find that there is a slight difference in the molecular structure of these two sugars. Now, the DNA structure, the way it is bonded together, the covalent bonds that form a nucleic acid, are very specific. If we look at this sugar here, you will notice that there are 5 carbons in it, and we can number them starting at the base. Carbon 1 right here, carbon 2 right here, carbon 3 right here, which is labeled, carbon 4 right there, which is not, and carbon 5, which is again labeled and attached to the phosphate group. The DNA is bonded together such that an hydroxyl group on the 3rd carbon bonds to the phosphate group of the next nucleotide, which is itself attached to carbon 5. So a hydroxyl group on the 3rd carbon bonds to the phosphate group, which is itself attached to carbon 5. This forms the backbone of the DNA. And as I said earlier, the nitrogenous bases are sitting off to the side. Well, here's the role of the nitrogenous bases. It's very important. Remember earlier in the year we said that certain lengths of DNA are the actual genes, and what they do is they code for a particular protein. Well, it is this sequence, this particular sequence of bases, and as you travel down the molecule, that sequence, which codes for the amino acids that make up the proteins. All right, now that's a pretty nifty system, and we're going to spend all of January learning about that. But for now, the important thing to notice is that we have a backbone with these nitrogenous bases, and each gene has a unique sequence of nitrogenous bases that specify what the amino acid sequence is in the protein that will be made from that gene. Now, you have all heard about the famous double helix that is formed by DNA. But what does that mean? Well, first of all, in the late 40s and early 50s, there was a great research chase to figure out what the structure of DNA actually was. And there were many research groups working on it. And finally, James Watson and Francis Crick figured out from X-ray crystallography that the DNA consisted of two strands of nucleic acids wrapped around each other like a spiral staircase, like the two sides of a spiral staircase, and that they were wrapped around each other in this double helix, helix being a coil, right? So that's the first important thing. You will notice that therefore there are two backbones, two sugar phosphate backbones on opposite sides with the bases lining up in the middle. Now the bases just don't flap around in the middle, but they actually only line up opposite particular bases. And you'll see that one of the purines is always across from one of the pyrimidines. One of the purines is always across from one of the pyrimidines. One of the purines is always across from one of the pyrimidines. But it's not just any purine and any pyrimidine. There's a specific way that the two have to be. Adenine always pairs with thymine, and guanine always pairs with cytosine. Now, that's interesting and very important, and we're going to find out more later about why it's important. But for now, the reason it's important is the reason this happens is because it maximizes the number of... What kind of bonds do you think these are here? Yes, you're right. Go ahead, say it out loud. These are hydrogen bonds forming between them. And remember, in water, when hydrogen bonds form, they're temporary. In DNA, when hydrogen bonds form, they're somewhat permanent. In other words, they're not a covalent bond, but they're always the nucleotides are always attracted to the opposite. The bases are always attracted to their complementary base pair. Thymine always attracted to adenine, and these bonds don't really come and go, except when it comes time to pull DNA apart. Why would DNA be pulled apart? Go ahead, see if you can think up a reason. There are two instances when DNA might be pulled apart. The two normal functions the cell has to do that might make it pull apart. Can you guess what they are? All right, well, here are the answers. One is when you're making a new copy of DNA. They have to be pulled apart, and the new copy has to be made. Two is when you're trying to read a gene. If you wanted to read, let's say, the gene that begins right here, you'd have to pull it apart and read this sequence of nitrogenous bases in order to know what protein to make. So we like to think of these hydrogen bonds as somewhat permanent, but in the same way that Velcro is. They're easily torn apart, and then they're easily put back together. All right, so here are some things you will need to know. First, you will need to know which base pairs are complementary. And the answer to that is adenine and thymine, or A, T, and cytosine and guanine, or C, G, right? Now, how can you remember that? Well, it's very simple. The only one that forms a word are complementary. AT is a word, at. I think you've heard of it. So that's the way I remember. The base pairs that are complementary are adenine and thymine. Here's the next thing you're going to need to know. Which bases are purines? And the answer to that is agriculture. Ag. Adenine, and guanine. And the next question is, how many rings do purines have? The way I remember how many rings purines have, it's the smaller word, and therefore it's the bigger molecule. They have 2 rings. Purines have 2 rings, whereas pyrimidines have only 1 ring. Now, as you can see, you have got a bunch of words that you now need to have in your vocabulary. You need to know adenine, you need to know thymine, you need to know cytosine, and you need to know guanine, you need to know purine, and you need to know pyrimidine. You need to know how to spell these words, and you need to know which ones are which. Okay, I'm back in the picture. I just wanted to show you now another way to look at DNA. Here is an example. Now look at every single one of these pairings. You see that there's always an A on one side, there must be a T on another side. There's a G here, there must be a C. If there's a C right here, what must be next to it? Yup, you got it, a G. If there's a T here, what must be across from it? An A. So that's the first thing. You need to memorize those base pairing, those complementary bases. The second thing I want you to notice is that the 2 backbones run in opposite directions. Remember earlier we had one where the 5 prime, which is the 5 carbon, is attached to a phosphate, and then it attaches down here through the 3 carbon to the next phosphate. So this one has the 5 carbon end up and runs like that, but the other strand has the 3 carbon end up, and so they run in opposite directions. That's called antiparallel, but that's one of the things you will need to know, that the 2 of them run in opposite directions. It becomes very important when we talk about DNA replication in January. Okay, in the email that I sent you, I also will include this document here, which is the little DNA quiz. And what I'd like you to do on this is to make sure that you can identify a phosphate in each chain, a sugar in each chain, and then I want you to label each amino acid, whether it's A, T, G, or C. And if you're having trouble figuring that out, one clue is to count the number of hydrogen bonds. And then I want you to point out which ones are purines and which ones are pyrimidines of the bases. Okay? So you think you can do that? And if you have any problems with that, give me an email. Shoot me an email. Okay, a couple of last-minute points. Here now we can compare DNA and RNA. On the left we have DNA. You can see that it is double-stranded. On the right we have RNA. It is single-stranded. On the left we have the 4 bases of DNA, the now familiar C, G, A, and T. And RNA's bases are the same, except for in place of thymine we have uracil here, a different base. And that's one way you could tell the difference between the 2 molecules, is that there would be a U only found in RNA. So just expanding what we've already learned into a slightly different realm to discuss something we've already discussed in this course. What you actually inherit from your parents is that actual sequence of nucleotides. That's what you get. And if we think about this in evolutionary terms, 2 more closely related species will have more similar DNA than more distantly related species. So we would expect alligator and crow to have more similar DNA than alligator and iguana or crow and iguana. So learning that sequence we can use to make these evolutionary trees that we learned about earlier in the year. Okay, so that's all you really need to know about DNA for tomorrow's test. Make sure you know the structure. I myself am getting ready to go back outside and have a snowball fight with my kids. I'm going to ambush them when they get home. And have a great afternoon. I'm going to go ride on my sleigh as well.